I have 4 minutes. I hope I make it. Hi, I'm Iris. I'm a bioinformatician in the functional genomics group in EBI. I'm going to talk about our two resources, which are Expression Atlas and Single Cell Expression Atlas. These are curated, reanalyzed multi-species knowledge bases for gene expression, and the Expression Atlas is mainly for bulk RNA-seq datasets and microarray datasets, which are old. There are also proteomic datasets in there. And Single Cell Expression Atlas is basically the single cell counterpart. So we ingest data from the research community by direct submission or by ingesting from the archives. We curate and annotate, and then the raw data and the curated metadata are then ingested by our production analysis pipelines and then loaded into the atlases. And once released, they are made available in the web interfaces of our atlases. So this is the Expression Atlas interface. You can search a gene and then also search or refine the search by including biological condition terms, which are also terms that are we maintain in a controlled vocabulary. And this is the results page, which gives you the experiments where that searched gene is expressed, and also the organism parts where they are expressed, and some tools to help you. For example, in this anatomogram, interactive anatomogram helps you visualize the localization and then also refine your search. This is a results page for differential expression experiments, and it also gives you a list of experiments where that particular gene is expressed. And if you click on any of these two results pages, if you click on an experiment, it takes you to the experiment page where you can do more exploratory analysis. And then also this interface is mirrored in a single cell expression atlas. Basically, you can also do a gene search, but aside from gene search, you can also do metadata search. If you do a gene search, then you're given a list of experiments where that gene is expressed. If you do a metadata search, then you're taken to a page where you have this cell type wheel, which is an interactive graphical representation of hierarchical metadata. And then you can refine your search using that cell type wheel. And if you click on the edges, which these are the cell types, then you can get these list the heat map, which has the experiments where the cell type is annotated, and also the marker genes, which are markers for the cell type and in each particular experiment. So if you click on an experiment, again, you're taken to an experiment page where you can explore that particular data set more. And so in the past year, we've also made some improvements in our production pipelines, particularly in single cell expression analysis pipeline, sorry. So one of those is ingesting externally analyzed data sets for projects that cannot actually give their raw FASTQ files. And so we have those externally analyzed data sets now in Single Cell Expression Atlas. And I'll skip that. We also have made improvements in our pipeline, in our analysis pipeline. Now, the old was Nextflow and Galaxy Hybrid, but now it is fully end-to-end Nextflow. We've updated the pipeline so that it's more in keeping with best practices in the community, and also more scalable, portable, and fair, fully modularized, and also no longer by production by default. Anyone can use the pipeline as is, as long as they have the correct input, and also the software, which is just software requirements, just Nextflow, and by default Docker. So that's it. Please use our resources, and also if you have data sets, please consider submitting to Annotator, which is our submission portal. And this is the team. Thank you.
Hi, thank you. I was wondering, how do you handle the fact that single cell dataset might annotate same cell type with slightly different names?
So we do annotation with ontology terms. So by the time they end up in our analysis pipeline, we would be using the ontology terms.
Okay, so you re-annotate the cluster in the
yeah.
Okay, thank you.